food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Laviolette, thank you for joining me again on the podcast for the second time.
Thanks for having me, Nick.
You want to just start off by reminding everyone who you are and what your lab studies?
Sure. My name is Steve Laviolette. I'm a professor at the University of Western Ontario. My lab is primarily interested in the cannabinoid system in the brain and how it relates to mental health. We're looking at various different models, including the impacts of exposure to cannabinoids during sensitive periods of brain development, like prenatal development, adolescent brain development. And we're also looking at modulatory systems of the cannabinoid system, including systems like the fatty acid binding protein system, as an example.
Mm-hmm, mm-hmm. And so I want to give everyone a sense for how lipids, fats, are used in the brain. As some listeners will know, but many may not know, the endocannabinoids themselves are an example of how fats are used in the brain for signaling rather than just structural purposes. But, when we think about endogenous cannabinoids, when we think about some of the lipid signaling molecules and related things we'll talk about, can you just give people a sense for how the brain uses fats beyond just structural components of membranes and things like that?
Right. No, it's an important question that's often overlooked because I think we tend to focus more on the neurochemicals and the neurotransmitters and sort of ignore the bigger picture. Fatty acids are really the major. The brain is mostly fat. It's mostly fat, obviously including myelination and a lot of fatty acid signaling molecules and whatnot. And beyond, of course, the structural importance of fatty acids, which make up most of the brain, the cellular membranes and whatnot. There's a lot of fatty acid signaling molecules in the brain as well. And of course, when they have to traverse through an aqueous substrate, like the intracellular milieu, for example, we need to have specialized proteins that can sort of chaperone those hydrophobic molecules across these aqueous membranes. So that's why we have a lot of different fatty acid signaling systems in the brain, including the endocannabinoid system that relies on these specialized proteins that allow the signals to get through to where they need to go in the brain.
Mm-hmm. Yeah. So, everyone knows sort of oil and water don't mix well together. Fats and aqueous things, watery things don't mix well together on their own. And that's why, I guess, another way to get into this and think about it is the same reason you sort of have these chaperone lipoproteins in your blood that shuttle cholesterols and fats around. We've also got various things in the brain that help us shuttle lipids around. And one of the reasons we do that the nervous system has to do that is because these fats aren't just components of membranes. They are signaling molecules that actually help the brain process and transfer information around.
Absolutely. And of course, they also serve as important precursors for a lot of different neurotransmitters. So they're involved in the biosynthetic pathways of a wide variety of neurotransmitters, including the naturally occurring cannabinoids in the brain.
Mm-hmm, mm-hmm. And so what exactly can you just give everyone a crash course in what the cannabinoids are at the molecular level and where they ultimately come from?
Right. So the endocannabinoids are, they're one of the most evolutionarily oldest systems in the vertebrate brain. And they're seen, across the entire evolutionary spectrum. And what they primarily do is regulate signaling in the brain. So they're primarily sort of retrograde modulators of neurotransmission. So, for example, the brain has to maintain a balance between excitation and inhibition. So the endocannabinoid system is very importantly involved in regulating inhibitory processes in the brain. So the presynaptic neuron will release endocannabinoids. And the two major endocannabinoids are called 2-arachidonoglycerol, or 2-AG, and then anandamide is the other major one. So those are the two major sort of naturally occurring endocannabinoids. They serve slightly different functions. We're still trying to figure out exactly what they do. But we know that both of them are involved in sort of this retrograde inhibitory modulation of the synapse itself, and primarily involved in sort of regulating excitation and inhibition balance in various brain areas. And of course, they're highly regulated in areas of the brain that are responsible for emotional processing, for cognitive processing, like the frontal cortex, the amygdala, for example, the striatum. So you can imagine that disturbances in the balances between those endocannabinoids are major risk factors for a lot of different mental health disorders.
Yeah. And I guess bird's eye view, the brain, there's information flowing through the brain in the form of electrochemical activity. That information needs to be patterned. It needs to be structured and constrained. And in simplified terms, you don't want overexcitation. You don't want underexcitation. And the cannabinoids are an important mechanism preventing things from getting out of hand or underhand.
Exactly. Yeah.
And so how does our body make them? Like, what are the pieces that go into producing them, and where do they ultimately come from?
Well, two of the major precursors for the endocannabinoids are DHA and EPA. I won't attempt to pronounce the full name, but those are, of course, the omega-3 fatty acids. And the omega-3 fatty acids primarily come from the diet. I'm sure you're aware that there's a lot of people are deficient in omega-3 fatty acids. They don't get enough in their diets. And of course, we rely on those omega-3s to sort of serve as the EPA and DHA to serve as the precursor molecules for some of these naturally occurring endocannabinoids. So it's a very complex biosynthetic chain. But ultimately, we do need to have those dietary precursors to get the adequate production of the endocannabinoids.
I see. So it's the omega-3s, not the omega-6s, that ultimately are the precursors to endocannabinoids?
The omega-6s also are involved in some of that pathway as well, but it's primarily believed to be the omega-3s that are sort of the major, especially DHA and EPA, which serve as the major precursors.
I see. And so when these things are used in the brain, so endogenous cannabinoids are famously made on demand. So as opposed to a lot of other chemical messengers in the brain, like neurotransmitters, they tend not to be packaged and sitting around waiting to be used. They tend to be made on demand. So are they basically just kind of clipped off of fatty components of the membrane?
Pretty much, yeah. I mean, it's a complex pathway, but they're primarily, you're right, they're sort of synthesized on demand, particularly when we're talking about mechanisms I was just referring to, like the necessity for retrograde inhibitory signaling across synapses and that sort of thing.
And so other than being retrograde messengers, what can you tell us about endocannabinoids that will sort of set us up to understand things like anxiety disorders and some of the other things we're going to get into in this conversation?
Great. So, both the naturally occurring endocannabinoids, 2-AG and anandamide, are critically involved in regulating emotional states, reward processing, aversive states as well, and of course anxiety levels. So we know that levels of anandamide, for example, and I primarily will focus on that because that's sort of been the focus of our work, anandamide primarily. Anandamide is very importantly involved in emotional regulation. If you have too little anandamide, that's linked to increased anxiety. But if you can somehow increase the levels of naturally occurring anandamide, that's been linked to decreased levels of anxiety as well. So there's a strong sort of balance between the levels of these naturally occurring endocannabinoids and sort of your baseline emotional state, your sort of state of anxiety. And that's been shown in both clinical and preclinical research as well.
Mm-hmm, mm-hmm. And one thing I want to connect here up front is, what can you tell us about the potential connections that the cannabinoid system may be involved in between things like anxiety and mood disorders on the one hand and things like neuroinflammation on the other? Is there a deep connection there between inflammation and mood regulation?
There is. I mean, that it's an emerging field, I think, trying to understand how neuroinflammation is involved in regulating mood and anxiety. We know that neuroinflammation can be strongly regulated by omega-3 fatty acids, for example. So, for example, the omega-3 fatty acids are known to decrease neuroinflammation. Omega-6s are more pro-inflammatory. So that's another component of what we study in the lab, is looking at the balance between omega-3 and omega-6 and how that controls neuroinflammation. And we've been able to show that that regulatory balance is also critical for sort of baseline levels of anxiety, not just anxiety and mood, but also cognitive behaviors as well. So, there is certainly a functional link between anandamide and neuroinflammatory mechanisms, but they go through a wide array of different signaling pathways. So, for example, they're linked to the PPAR system, which is the peroxisome proliferator activation receptors. They're linked to nuclear transcription of various intracellular components that are involved in the inflammatory state of the cell. But sort of, broad perspective is that they're definitely involved in regulating neuroinflammatory mechanisms, and disturbances in those neuroinflammatory processes have definitely been linked to mood and anxiety disorders.
Mm-hmm. So the omega-3s and the omega-6s, these are long-chain polyunsaturated fatty acids, which, for people that don't think about lipids a lot, they're long molecules. They're usually bendy because they've got multiple double bonds. So they've got, particular structures to them that give them the functions they have. And, as you've written in this review paper I looked at before meeting you today, and as I've discussed in other contexts on the podcast, one thing that seems to be important about the omega-3 and 6 fatty acids is that they're similar to each other, but they're functionally divergent. In fact, they often kind of do opposing things, and you can't convert one into the other. And so this brings us to the concept of basically dietary balance, that these things are supposed to be in relatively comparable balance because they often have, say, pro-inflammatory effects on the one hand versus anti-inflammatory or resolving effects on the other. What can you say about that in terms of sort of dietary PUFA intake and this concept of the balance between the omega-3s and the omega-6s?
Right. Well, I'll frame that in some of our other research. So we've been looking at primarily the impacts of prenatal THC exposure. So we published a couple of papers recently looking at how THC exposure, which is obviously an extrinsic cannabinoid, but that can strongly throw off the balance of the omega-3s and omega-6. So in a recent paper that we published in Molecular Psychiatry, we found that exposure to THC during pregnancy using an animal model, it throws off the balance of omega-6 and omega-3 in the brain. And so we did imaging of those pathways in the brain and showed that when that balance was thrown off, regardless of it, and again, they sort of have to be, you want to have sort of an ideal ratio between omega-6 and omega-3. When that balance gets thrown off, the offspring of those animals that have been exposed to THC, they had problems with cognition, they had problems with mood and anxiety symptoms that persisted all the way into early adulthood. So, so I was sort of framing it in that light. What that indicates is that you do need to maintain that sort of healthy balance between omega-3 and omega-6 throughout the entire lifespan to make sure that any sort of psychopathology is held in check in that respect anyways.
So you expose pregnant rodents to THC, it throws off the omega-3, 6 balance, and then the offspring end up having effects down the road. Can you unpack that a little bit more for us? What was the direction of what's the normal balance at baseline between the omega-3s and 6s? Is it roughly 1-to-1? And then in which direction did the THC push things?
Ideally, I mean, it sort of depends on the species we're talking about. And rats generally want to have a bit more omega-3 than omega-6.
Okay.
And this did the opposite. So this up the levels in most of the brain areas we looked at, up the levels of omega-6, decrease the levels of omega-3
within the brain.
And I'm sorry.
Within the brain.
Within the brain, yes. So we've actually developed an imaging system to look at these fatty acids in the brain.
Wow.
So we can actually visualize the levels of and quantify the levels of the fatty acids, not just omega-6 and omega-3, but all of their sort of downstream signaling pathways as well. And so what we were able to show was that ultimately, we see this disbalance between and we see an upregulation of omega-6, downregulation of omega-3, and we have more recently been able to show that a dietary intervention with omega-3 fatty acids was able to prevent a lot of those negative impacts. That paper is coming out in a couple of weeks.
I see.
And so, yeah, so that's really been a focus recently. And that was, of course, a dietary intervention that we gave the pregnant rats while they were being exposed to the THC. And we were able to block a lot of those negative impacts that was protective all the way into early adulthood.
I see. So it sounds like sort of the two stories that have emerged or are emerging are, if you give a pregnant rodent THC, it throws off the omega-3/6 balance. It skews it towards omega-6s. This has long-lasting consequences, negative brain effects, basically, for the offspring down the road. But it sounds like what you're saying is the new story coming out is that if you do that, but then you also maybe supplement with omega-3s, you can mitigate that effect?
Exactly. Yeah. So that's the paper that's coming out. And it was a specialized diet that we had prepared, and it was a high omega-3 diet. And basically what we were able to do was prevent a lot of those negative impacts of THC. There's sort of this misperception that cannabis during pregnancy is safer than other drugs that other people that people may use to mitigate some of the negative impacts, some of the negative symptoms of pregnancy. But the research that is emerging is showing that they can have profound effects on those pathways. We don't know exactly why this is happening. That's important to keep in mind because it's such a complicated biosynthetic pathway. We think that there's probably some sort of competition going on. If you're flooding the brain with these extrinsic cannabinoids, that might be competing with the endocannabinoid system, anandamide and 2-AG, for example. And that's throwing off the normal regulatory pathway and ultimately throwing off skew the balance between omega-6 and omega-3. And importantly, these disturbances were in brain areas that are linked to very serious mental health problems, like the prefrontal cortex, the hippocampus, the striatum. And they're linked to all sorts of risk factors for various problems in later life.
Yeah. One thing I want to ask you, just as kind of a connoisseur of this area myself, is a bit more detail here about something that I've had Margaret McCarthy on the podcast before, and she does really cool work, in my opinion, that has to do with the effects of cannabinoids on development, and part of her world involves the fact that these things often have sex-dependent effects. Did you guys do these studies in male versus female mice, or see anything interesting there?
We did, yeah. I'm glad you asked that. We saw massive sex differences.
Really?
It was quite remarkable, actually, and even the brain areas that were impacted were different. So hopefully I'm getting this right, but essentially, we were primarily focused on the prefrontal cortex and the hippocampus, which are obviously very important for disorders like mood anxiety disorders, schizophrenia, cognitive problems, whatnot. And what we found was that the impacts on these fatty acid pathways were different in the males than the females. The males had more profound deficits in the frontal cortex. The females had more profound problems in the hippocampus. And that was reflected in the behavioral symptoms as well. And we don't know exactly why that is, because obviously the developing male and female brain are going to be exposed to different hormone levels and whatnot. We expect it's probably something to do with the fetal milieu. But we also work with our collaborators at the Western University, Dan Hardy's lab, and he's looking at the way we sort of conduct the research is that Dan Hardy's lab looks at the downstream physiological effects, like on the liver and on the placenta. We published a paper a couple years ago where we showed that the prenatal THC exposure disturbs the transfer of fatty acids, the vascularization of an area of the placenta called the labyrinth zone. And the labyrinth zone in the placenta is necessary for sort of that nutrient exchange between the mother and the developing fetus. So we think it's starting in the placenta. And but ultimately the impacts on males and females is profoundly different. And this persisted all the way into early adulthood. So depending on whether we're talking about a male or female fetus, there's going to be some very different effects. And that has implications also for intervention strategies, because obviously the frontal cortex and the hippocampus are very different. They subserve very different functions in the brain and are associated with different mental health disorders. So that's sort of the next step in the evolution of our research. We're trying to figure out how these intervention strategies, like for example, a dietary intervention strategy, can that differentially impact the male versus female developing brain? It's a really important question.
So you saw big effects from THC exposure in pregnant females on their offspring, whether it was males or females, but the effects were just different in each sex.
Yes, absolutely. Yeah.
Wow.
Yeah. Even we were surprised by that. We expected some differences, but nothing that profound. So we were. Yeah.
Okay, so some of these things can be affected by dietary interventions. It sounds like, and we know, I think, that THC is a CB1 receptor agonist, and CB1 receptors are located all over the place, not just in the brain. I believe they do have direct vascular effects, so that would kind of start to make sense of why you would see some kind of placental exchange phenomenon happening.
Yeah, exactly. Yeah. I mean, it's a ubiquitous system throughout the entire body. So we sort of, I'm obviously biased towards the brain, but of course, there's a whole sort of peripheral physiology associated with the cannabinoid system as well.
Yeah. And as you mentioned, sort of the biochemistry and the complexity here is really immense, especially when you're talking about these lipid synthesis and exchange pathways and all this stuff. So it can be hard to pinpoint exactly what's going on here. But for me, when I sort of try and distill a lot of this complexity down into something like a general principle, we know that the omega-3s and 6s the system is evolved. They're set up in a way that, again, they can't interconvert. They're competing for access to the same enzymes. The balance of them is very important. And so no matter what the details and the minutia here are for how THC is doing these things, it is somehow throwing off the balance of lipids so that it's outside the normal range of what development is sort of expecting to happen.
Exactly. Yeah, that's right.
Interesting. And so one of the things I want to talk about too is these things called fatty acid binding proteins. So as you mentioned in the beginning, fats are very important. They're not just structural components. They're used as signaling molecules in the brain. But there's this sort of challenge that our biology has is, okay, we're an aqueous sack of stuff. We're very watery. Yeah. So how do we move around fatty things? And one of the ways that fats get moved around is there's special proteins that kind of shuttle them into and or out of cells. So what can you tell us in basic terms about these fatty acid binding proteins and how they work on a high level?
Yeah, it's a fascinating system. And it's something that we've only, the past couple of years, we've sort of been getting into this, primarily because what's remarkable about them is that it's a system that if you pharmacologically manipulate it, you can bypass the traditional cannabinoid receptor system. And the way they work is that they essentially chaperone or piggyback these endocannabinoids in the aqueous environment and bring them to their degradation sites. And in this case, we're talking. Let's take anandamide, for example. Anandamide levels need to be tightly regulated, just like any other molecule in the brain. And the way that's done primarily is through fatty acid amide hydrolase, or FAAH, which is becoming increasingly well known as a modulator of the endocannabinoid system. So essentially what the FABP proteins do is they chaperone the anandamide to its degradation site and allow it to be degraded and hence control its availability. We've identified 10 of them so far, and they serve, again, a wide variety of different functions, not just in the brain, but the body as well, as would be expected. But in the brain, it's primarily FABP3, FABP5, and FABP7 that are the best understood. There may be more down the road, but those are the ones that we've identified recently. We've been working with a FABP5 inhibitor, and what we were able to show based on what we know about the role of FABP5s is that if you pharmacologically inhibit FABP5, you can cause anti-anxiety and antidepressant-like effects in various brain areas. And the hypothesis is that that's because we're increasing levels of anandamide in the brain. Because if you're taking away that system that shuffles it to its degradation
site,
you're going to increase it. And there's a wide variety of preclinical studies using mutant mouse models and we talk about this in the review paper, where they knock out these various proteins, whether it be FABP3, FABP5, or FABP7, And almost sort of the universal outcome is that you see these elevated levels of anandamide. And that's also associated with anxiolytic effects or anti-anxiety effects. And our lab has also shown it helps to buffer against the effects of chronic stress. And it has acute anti-anxiety effects. And what's really interesting about the system, and the reason we started getting into this, the signaling system, is because it can bypass it's not directly activating the CB1 or CB2 receptors.
Yeah.
It's an indirect way of modulating them, because obviously if you take THC, for example, or cannabis, what have you, you're activating CB1, and that's associated with a host of side effects that you don't necessarily want. And of course, some people actually experience acute paranoia and anxiety if they activate the CB1 receptor system. So it's not really an ideal target for treating anxiety, even though some people do get, antidepressant and anti-anxiety effects. Yeah. Other people, it's sort of a dose-dependent thing, right? So depending on your sensitivity to it, you can actually have the opposite effect. So we were interested primarily in using this. It was a proprietary compound we used. We work in collaboration with a company called Artello Biosciences. They're based in San Diego, and they've developed these proprietary FABP inhibitors. And so they, ultimately we wanted to test them in terms of their anxiolytic effects. And we've published a couple papers already. We talked about them in the review paper. There's a few more papers on the way. And we're quite impressed with their anxiolytic and antidepressant properties. And what's really great about them is that, at least in our preclinical models, they don't seem to have any addictive liability. So they don't seem to modulate, for example, the dopamine system. Most drugs of abuse will activate dopamine. These compounds don't seem to activate dopamine. They don't seem to produce any negative cognitive effects. So based on our preliminary preclinical work, they seem to be a very safe sort of alternative to more traditional compounds. And more importantly, as I'm sure you're aware, about 13% of the US population uses benzodiazepines in any given year. And of those people, 2% will develop a very strong dependency on these traditional anti-anxiety medications like the benzodiazepines. And it's a very, very devastating addiction associated with very profound withdrawal symptoms and even longer term physiological side effects that you don't want. So again, what we think is that modulating anandamide through this FABP inhibition system can serve as a safer anxiolytic or antidepressant alternative to the more traditional medications that are out there, and also to cannabis.
Yeah. Interesting. So again, taking anandamide as an example, the brain is using anandamide. It's secreting it to do normal stuff in the normal course of biology. One of the ways the brain regulates stuff and how much of it is around and what's happening is, it'll release something like an endocannabinoid or a neurotransmitter or what have you, and then it will tightly regulate how quickly those things are sort of cleaned up and degraded. And that prevents them from, diffusing around, sticking around for too long, it's just a form of regulation. So anandamide gets released and then it gets degraded. And that's an important part of its regulation. A key piece in this puzzle for how it gets degraded in this manner is that you've got these fatty acid binding proteins that are basically grabbing it and shuttling it to where it gets chopped up.
Exactly. Yeah,
got it.
That's exactly how it works. Yeah.
And so when you have a FABP5 inhibitor in this case, so instead of a drug that is going to say bind to a receptor like the CB1 receptor and activate it or inhibit it, this is binding to the shuttling protein. And this, you're basically saying this seems to have certain advantages. And the way I'm hearing this is, if you have a drug that binds to a receptor, and let's just use CB1 as an example, it might bind to that receptor and activate it. It's going to compete with endogenous ligands. It might cause some desensitization or some tolerance, what have you. There's going to be all of these effects that are secondary, what we think of as side effects, to the primary effect of it actually engaging the receptor. When you have something like this FABP5 inhibitor, it's not directly engaging the receptor of anandamide. It's just regulating its degradation. And so you're sort of not upsetting the natural pattern of release of anandamide in this case.
Exactly. Yeah. And we think that's largely the reason why it has a better safety profile, because it's not modulating the cannabinoid system. What's also interesting that I mentioned is that most of the effects we've seen in terms of its ability to induce anti-anxiety effects or antidepressant effects, it actually seems to be CB2 dependent. So, which was also very surprising, because naturally you think CB1 is more ubiquitous than CB2. We don't really understand exactly what CB2 is doing. We know it's linked to like astroglios and that sort of thing, astrocytes, and some neuroinflammatory mechanisms, mitochondria regulation, for example. But that was quite interesting. So when we tried to block the effects with a CB1 antagonist, there was no blockade, but the CB2 antagonist blocked it. So to sort of step back, what we think is happening, and this is subject to change, but what we think is happening is that the FABP5 inhibitor is blocking that chaperone, so we're getting increased levels of anandamide. So where does the anandamide go? We think it's going to CB2 to produce these behavioral effects, these anxiolytic effects, and that's another piece of the puzzle we still have to solve, because CB2 is such a mystery.
Right.
So we're trying to figure out exactly how CB2 and or its downstream pathways might be related to its actual systems level effects.
Well, it sounds like there's an important lesson there, which is, this is more general than the area of biology we're talking about. You can look at many areas of biology and see this type of thing, I think. But, when you first start learning about the endocannabinoid system, you often learn CB1 is more abundant in the brain, CB2 is more abundant in the periphery. And therefore we start to think, if we get sloppy, CB1 means brain stuff, CB2 is not important in the brain. But what you're saying is CB2 is important, even though it's not nearly as ubiquitous in the brain. That doesn't mean it's not doing anything substantial, because in this case, you found an example where it seems to be.
Exactly. And that was the dogma for decades, right? It was CB1. And the dogma for decades was there were no CB2 receptors in the brain. They thought it was just, and that's fair enough. It's only been, a few decades that we've actually even, sort of like characterized the cannabinoid receptor system with the studies that were done way back in the 60s. But having said that, you're absolutely right. I think it's really critical that we consider both, the CB1 and the CB2 system. Obviously, CB2 does have some functional importance in the brain, but and the other, and of course, how it's impacting other neurotransmitters as well. Because as I mentioned at the outset, both CB1 and CB2 are ultimately sort of modulatory systems, right? So they help to regulate the release of other neurochemicals, things like serotonin, dopamine, GABA, glutamate. So how are they modulating those different, more traditional neurotransmitter systems, which we know are very importantly involved in anxiety and depression and addiction, for example.
Mm-hmm. And then, what can you tell us more about? So I think you said, there's a bunch of fatty acid binding proteins, maybe 10 of them that have been characterized in mammals. And, like many things where there's multiple versions of it, one copy might be strongly expressed, one version might be strongly expressed in the brain, another in the liver, and so on and so forth. These things are going to serve slightly different function in all of these different tissues. I think you said FABP5 is one of the ones that's strongly expressed in the brain. Is that true? And what can you start to tell us about, what happens when you start messing with FABP5 in a normal animal? What goes wrong when it's missing, for example?
Yeah, well, if you knock it out, actually, if you knock out those proteins, you actually tend to get anxiolytic effects. So similar to pharmacological modulation. The problem, these animals do have other abnormalities, though. There's some studies have shown that they've got other disturbances, and not just FABP5, but FABP3 and FABP7 as well. But of course, it's sort of apples to oranges, because when we're talking about these mutant animals, they're getting the entire system wiped out from birth, right? There's all these sorts of compensatory systems that kick in to sort of compensate for that loss of that system. And that's one of the sort of the shortcomings of that technology, and using those knockout technology. So there are definitely some side effects from wiping out the entire system from birth, which you would never see in the natural world in any case. So that's why I think it's probably from a clinical perspective, it's better to take more of a pharmacological approach. And so what we've done in our research is we started out by targeting brain areas that we know are involved in anxiety and depression. So, for example, we actually started out doing microinfusions of the FABP5 inhibitor in the prefrontal cortex. Then we follow that up with microinfusions into the amygdala. What we found with the prefrontal cortex was that not only did it produce acute anxiolytic effects in these rats, these were adult rats, but it also inhibited the output of the amygdala. So the amygdala and the prefrontal cortex are connected with each other. We've studied that pathway for many years now. And so they can excite each other or inhibit each other. And what we found was that, just as an example, the FABP5 inhibitor in the prefrontal cortex could sort of blunt the output from the amygdala, which of course is like one of the primary anxiety centers of the brain. If you have an overactive amygdala, you're much more likely to have, symptoms of anxiety disorders, that sort of thing. So that's just one example of sort of a more targeted way to map out the functions of FABP5 as opposed to just sort of a universal knockout mutation.
Yeah. And, some of the results that you mentioned earlier, you could mitigate the effects of THC, for example, with a dietary intervention. What do you think more generally about, what does that say about the potential potency of making dietary fat changes on brain development generally?
Yeah, I think it's sort of a neglected area because we're certainly guilty of this as well. I think a lot of sort of neuropharmacology research programs focus mostly on traditional neurochemical pathways and small molecules and drugs. We're ultimately looking at pharmacological manipulations. But what if we can do, just simple dietary interventions? And that's what we were able to show with the other paper as well. There are some really exciting clinical studies out there. Like there's a very famous clinical study called the Vienna omega-3 study. And what they did was they gave, I believe it was a 6-week dietary intervention with a high omega-3 diet. And they gave these to a population of people that were at high risk for converting into full schizophrenia. And they were able to show that just that simple dietary intervention for 6 weeks was able to strongly decrease that conversion rate into psychosis in these people. So, but the data is mixed, right? I mean, there's some positive findings, some negative findings. I don't think we fully understand why some of these studies have been positive, some have been negative. We know that there's some evidence that if you combine traditional, for example, schizophrenia medications with dietary omega-3s, for example, you can in some cases enhance the clinical impacts of those pharmacological interventions. So I think there's a huge opportunity to look at how dietary interventions can modulate not only sort of the baseline risk factors, but also can it improve the efficacy of some of these drugs that we use, ad hoc. Can they improve the efficacy of some of these other compounds that we've been used that we know have negative side effects?
Mm-hmm. How do these fatty acid binding proteins tie into things like the HPA axis and stress hormones? Do they have an effect there?
I don't know. Off the top of my head, I couldn't answer that too well. We haven't studied that ourselves. I would certainly expect that given its anxiolytic effects, very strong anxiolytic effects, you're going to ultimately regulate cortisol. It's funny you asked that because we're actually starting some studies now where we're going to be measuring cortisol levels following exposure to FABP5 inhibitors, but we don't have the data yet. So I will have an answer in a few months. But my hypothesis is that it would definitely decrease levels of corticosterone and cortisol, for example, because they are associated with high anxiety states. I couldn't tell you what the exact mechanism is, but that sort of at a broader level, I would expect that to be the case.
Mm-hmm. Okay, so FABP5, fatty acid binding protein 5, strongly expressed in the brain. This can grab an anandamide, bring it into the cell where it's then degraded. So more FABP5 would mean faster anandamide degradation and lower anandamide levels, which is going to tend to be higher anxiety. And then if you inhibit, you say a FABP5 inhibitor, then it will shuttle in anandamide less. It will be degraded more slowly. That will tend to elevate anandamide, and that's going to be correlating with anti-anxiety effects.
But yeah, and antidepressant as well. I'd add that as well, because we've also been doing some studies looking at chronic stress. And so again, these are preclinical models, of course, but obviously the number one cause of mood and anxiety disorders is stress. And it's usually a long-term stress that sort of leads people down that clinical path. And so we've been looking at that as well. And, we do have some promising preliminary evidence that the FABP5s can sort of mitigate the impacts of chronic stress as well. One area that we've been looking at is the hippocampus and neurogenesis. So this is data that we've only presented sort of at a preliminary level about essentially what we've been able to show is that FABP5 inhibition can reduce the reduction in neurogenesis that is associated with chronic stress. So if you're under a lot of stress, your hippocampus stops making new neurons, which is called neurogenesis. But what we've been able to show at the preliminary state is that the FABP inhibition can block that process from occurring and allow the hippocampus to continue to produce new neurons, which is correlated with an antidepressant-like effect.
I see. So most parts of the brain, most of the time, aren't doing neurogenesis, aren't making new neurons in an adult mammal, but there's a couple special parts of the brain where that does continue to happen. The hippocampus is one of them. And so these things are having an effect on, so chronic stress tends to decrease neurogenesis, basically anxiety, stress, bad stuff tends to decrease it. And then you see the opposite effect in the opposite direction. You're seeing that the decrease in neurogenesis that comes from stress is more mild when you mess with these fatty acid binding proteins.
Exactly. Yeah.
Interesting.
So that's again, another mechanism. And there's probably others as well. But of course, we've focused primarily on the hippocampus and the prefrontal cortex. But I would imagine there might be similar mechanisms happening for other areas as well. But of course, the neurogenesis, as you mentioned, neurogenesis is really in adults is limited primarily to the hippocampus.
Yeah. So you got multiple fatty acid binding proteins, roughly 10 of them in mammals. FABP5 is the one we're talking about in the brain. FABP5 binds to anandamide and shuttles it in. That's what we were just discussing. Coincidentally, I was talking to some people on the podcast not too long ago, focused on a different area of biology and a different tissue, and they were also focused on FABP5. And long story short, what they discovered is that breast cancer cells actually sort of overexpress FABP5, and they shuttle in linoleic acid, which is the most abundant PUFA, polyunsaturated fatty acid found in seed oils, and it, connected to mTOR and drove cancer, and that was a separate story. The question I have for you is, okay, if FABP5 is highly expressed in the brain as well, and it's shuttling in anandamide, I'm guessing in the brain it can also grab onto linoleic acid. What can you tell me about these FABP5 proteins, and they're obviously not specific for one fatty acid. Are they able to grab a bunch of different ones? And how does that start to play into this?
Yeah, I mean, it's a huge list. I mean, it's not, I wouldn't want to give the impression that we're just talking about anandamide. It's involved in other endocannabinoid signaling molecules as well, and other fatty acids, including linoleic acid. We've never looked at that specifically. And I'm not, off the top of my head, I'm not aware of any study in the brain. And forgive me if I missed the data, but I'm not aware of any sort of similar comparable mechanism with linoleic acid in the brain. But that's very interesting. What you mentioned about breast cancer. I know that there are our industry partners we're working with are also running some clinical trials on cancer as well. Unfortunately, I'm not. I don't know too many of the details of those studies because we're focused on the mood and anxiety side of the story. But there are some promising findings in the cancer sphere as well, and that's probably related to similar mechanisms.
But these FABP5 proteins in the brain, they're probably affecting the shuttling of everything from the endocannabinoids to other dietary fatty acids, and they're grabbing onto many different things.
Absolutely, yeah, and that's an important thing to keep in mind as well. The off-target effects. There's always going to be a host of off-target effects, and that's part of the reason why we run all of these control studies. So, for example, if there was a risk that modulating other fatty acid transport with inhibiting FABP5. What if one of the side effects was to produce dependence or cognitive deficits? So that's why we run those kinds of control studies to sort of rule that out as a potential off-target side effect. There's a lot more work to do, but I think that's the given the complexity of not just FABP5, but all of the fatty acid binding proteins and what they're responsible for chaperoning in and out of the cell into different degradation sites. It makes the research very challenging because you have to account for these potential off-target side effects as well.
And when we think about these FABP5 inhibitors, I know that a lot of work has been done over the years trying to, get FA inhibitors to market. The endocannabinoid system is very important for many things. It ties into anxiety and depression and mood regulation, as we've discussed. FA inhibitors have been the object of study for a lot of companies over a lot of years. Yeah. Can you give us just a sense for in terms of endocannabinoid system-based drugs for mood regulation, what has worked, what has not worked, and sort of where does that whole area sit right now? Are people still excited about FA inhibitors? Are these FABP5 inhibitors the next thing? What's that look like?
Well, you're probably, I'm sure you recall the tragedy that happened with, there was a clinical trial with a FA inhibitor. I believe it was an irreversible FA inhibitor, and I believe there was at least one fatality. This was several years ago. At the top of my head, I don't remember the name of the drug. I think it was in France. This happened, and there was a fatality associated with the use of the FA inhibitor. So I think that did sort of dampen the enthusiasm, because you're right, it was a very 10 years ago, it was a very hot area, the use of FA inhibitors. I know there's certainly still interest in it, and there are still companies that are looking at the potential for FA inhibitors, primarily the reversible FA inhibitors as opposed to the irreversible FA inhibitors, because the irreversible ones appear to have the worst side effects, including things like suicidal ideation, gastrointestinal side effects, that sort of thing. So there are a lot of off-target negative impacts associated with FA inhibitors. But to answer the question, I think there's still some interest in that. Probably more interest in the FA inhibitors than FABP inhibitory molecules. But yeah, I think it's just sort of something that's going to have to play out with clinical trials. And I'd raise that point also, that ultimately we'd like to move ahead with some clinical trials to translate what we've done in the rodent studies into actual populations that are suffering with mood and anxiety disorders, to see if there's actually a translational benefit that we can observe with clinical populations.
And so if exogenous cannabinoid exposure during pregnancy can lead to developmental abnormalities that manifest in cognitive and mood problems, and if diet can mitigate that effect, does that also mean that simply being mindful of diet is sufficient to cause or maybe prevent things that we might consider mood or cognitive disorders?
Yeah, that's a great question. I mean, I think the role of diet is largely ignored in most modern neuroscience. I think nutritional neuroscience is a field that really needs to be elevated, because we've certainly, I'll talk about a few other studies we've looked at. So not just omega-3s, we've been looking at antioxidant interventions. We published a few papers with L-theanine, for example, N-acetylcysteine. These are both antioxidant compounds. You can go into the health food store and just pick your drugstore and just pick these up. They're very well tolerated. They have very few side effects. And what we've shown in the preclinical studies is that the antioxidants can block a lot of the negative psychiatric effects of cannabinoid exposure. Primarily during most of our studies have focused on adolescent exposure. So, as you're no doubt aware, there's a huge clinical correlation between adolescent cannabis use, especially high potency cannabis use, and the risk of schizophrenia. So we published a couple papers recently looking at both L-theanine and N-acetylcysteine interventions to see if they can mitigate the negative impacts of THC, just like I was talking about before with the omega-3s. And they were very promising. They were able to block a lot of those negative effects. Again, we think this is related to oxidative stress caused by THC.
Yeah.
And that's also linked to neuroinflammation. the two are tightly linked to each other. And so what we're seeing, sort of the story we're seeing emerging, is that, it's probably oxidative stress and neuroinflammation that is the primary driver of a lot of the pathological risk for extrinsic cannabis exposure. So especially high, and I want to emphasize, we're talking primarily about like high potency THC.
Yeah.
Now, having said that, there's also been some studies that have come out recently with CBD, which has largely been sort of touted as this miracle cure-all for many years now. And there is some concerning preclinical evidence suggesting that maternal CBD exposure can lead to some negative long-term side effects as well. So again, just sort of the work.
Yeah. What is that? What is known there about CBD?
Yeah, so there's evidence that CBD, maternal CBD exposure, so prenatal CBD exposure can cause a lot of the similar symptoms as THC. So cognitive problems, emotional dysregulation, and they're primarily preclinical rodent models that have looked at that. But I know that there's a few that have come out, and we also had a paper recently where we looked at THC, CBD, and the combination of THC and CBD during prenatal exposure, and in many cases, the CBD and THC were producing equally negative effects.
Really?
Sometimes the combination would decrease the effects. Sometimes they would synergize the effects. So it's a really complicated story that's emerging. But suffice to say, this idea that CBD is this naturally safe and effective option for dealing with some of the negative symptoms of pregnancy, I think that is a myth that is being dispelled by a lot of this research.
I see. Interesting. And I want to touch on something you mentioned there. So you mentioned that THC can induce oxidative stress and have inflammatory effects. Now, some people listening might find that a little confusing, and I want to unpack and emphasize this for people because I think it's important. There's plenty of other contexts, right, in which you can go in the literature and say THC is anti-inflammatory. We know that many cannabinoids, endogenous and exogenous, are. But I think what we need to talk about here is the fact that something can be anti-inflammatory and pro-inflammatory, because inflammation is not one phenomenon driven by one mechanism. There can be ways that anti-inflammatory effects are driven by a cannabinoid like THC, for example, and other ways in which it drives, say, oxidative stress. The difference there could be dose and frequency. The difference there could just be parallel mechanisms that have different effects in different places. Can you just sort of unpack that for people? Yeah,
well, as you mentioned, I mean, one of the complicating factors is the number of routes of administration. So their edibles are becoming more popular. And we know the edibles obviously have a different metabolic response in the body, and the amount that gets to the brain is different with smoking it versus eating it. And then there's also very high potency THC extracts like shatter, for example, which that can be almost 90% THC that people are taking. So the general idea is that if you're taking a product that has roughly equivalent THC and CBD, you're mitigating some of the negative impacts of THC. They're sort of balancing each other out. But we know that, and I'm speaking from the Canadian perspective, where cannabis is, literally on every corner now. The most popular cannabis products contain 25 to 30% THC and like almost no CBD. So we're talking about high potency THC products that are the ones that are the most popular in the marketplace. So we think those are probably the primary culprit for inducing some of these sort of oxidative reactions or neuroinflammatory reactions. But again, we don't fully know the answer. And there could be things that are happening at the level of the liver, even that are altering how these effects play out in the brain. Even the microbiome. We've started doing some microbiome studies as well, and the data is too preliminary to talk about really. But we do know that edible cannabis exposure can alter the gut microbiome, which is critically linked to, I'm sure you're familiar with the gut-brain connection there. So we know that a lot of neurotransmitter precursors come from the gut. And there's this crosstalk between the gut and the brain. So I think that might have a lot to do with it. And that's, again, going to be wildly different if you're eating it. And so if you're taking edibles or you're smoking it, smoking is going right, largely right up to the brain, right? Going right into the bloodstream. Whereas edibles take a much longer time, much sort of a slower tonic reaction. So I think that has a lot to do with it. The potency, the route of administration, and of course, individual differences. So everyone responds a little differently to cannabis. Not everyone has the same reaction. So, genetic predispositions. We know that certain genetic predispositions make you more likely to suffer from psychiatric side effects of cannabis exposure. And we're just sort of unraveling that now. We're doing a lot of RNA sequencing in our lab. So we're looking at how cannabis exposure can alter the transcriptome of the brain, and what sort of transcriptomic changes are induced by cannabis exposure. One of the other models we've moved into recently are organoids. So we're taking basically growing mini brains, which I'm sure you've heard of. So you basically take an iPSC cell from a patient. So we have iPSC cells from patients with schizophrenia, and we take that cell and grow it into a mini brain, right? An organoid. And
you take stem cells from a tissue sample, and it grows into a mini brain that, on the one hand, has a lot of structural similarities to a real brain that's in a person, but because it's in a dish, you can interrogate it more finely and in more detail.
Exactly, yeah. So it's sort of a nice intermediate step between the rodent work and actual clinical trials, because we're dealing with real human tissue, real human cells. It's not a perfect model by any means, but it can replicate like cortical structure, for example. And this is not published data, but what we have done so far is looked at different combinations of THC, CBD, THC, and CBD in a dish. And again, what we're finding is that there are profound alterations in those fatty acid pathways, DHA, ARA, arachidonic acid, and other signaling pathways. It alters various neurotransmitter pathways as well. And some preliminary RNA sequencing, and this is very preliminary data, it's showing that it activates a lot of neuroinflammatory pathways. So the story is all falling together, and it's showing that most of the pathological risk factors, we think, are probably linked to these oxidative reactions and these neuroinflammatory responses.
Okay, so two things I want to emphasize here for people. So one, you mentioned, a few minutes ago that there's this notion out there that THC and CBD are the opposite. THC gets you high, and CBD is sort of like the safe, tame one. And in some ways, that is true. In many ways, THC and CBD do have functionally distinct and almost opposite effects at the level of, say, the CB1 receptor and things like this. But it's not that simple, unfortunately. You mentioned that there's data now showing that CBD exposure in the womb can cause problems that are directionally similar to those caused by THC. And so it's unfortunately not going to be the case that we can just think of, CBD canceling out the effects of THC or vice versa. And then the other thing is, you mentioned that THC can have these inflammatory or oxidative stress inducing effects. And even though it's true that THC can have anti-inflammatory effects, it can bind the CB2 receptor, it can do those things. The specific way it's ingested, the specific way it's processed, these ultra complex lipid signaling systems that are being tapped into here mean that, it's probably going to cause both lower and higher inflammation and lower and higher oxidative stress in a dose-dependent manner, in a form factor-dependent manner, in a exposure length-dependent manner. And it's unfortunately not going to, biology is not so simple that you can just say something like THC is anti-inflammatory, period.
Yeah, absolutely. And the story keeps evolving. And when we sort of first started dissecting out CBD and THC, we were studying them independently. And, we were able to show that they went through very different neurochemical pathways. So, for example, CBD primarily produces its effects through the 5-HT1A serotonin receptor, at least in our hand. And there's evidence from other labs as well showing that it modulates serotonin signaling as well. Whereas THC primarily seems to cause a dysregulation of dopamine. And then another study, we were able to show that CBD could actually block hyperdopamine induced by chronic amphetamine exposure, which is a preclinical model of schizophrenia.
Yeah.
So the story that was emerging was that they were wildly divergent in their effects. CBD was the good guy, THC was the bad guy. You know, very simplistic. But now we know when we started combining them, for example, or looking at different doses, then you start seeing these more subtle. It's not so simple. CBD, well, you go back to the idea the dose is the poison, right? So, depending on the dose of CBD, we could see different effects. And depending on the combination of CBD and THC, sometimes CBD can mitigate the negative impacts of THC, but sometimes they seem to actually produce worse effects when it's almost like CBD is unlocking some of the negative effects of THC, and we have no idea why. We don't know exactly what's happening, and I think ultimately it's going to come down to what brain area we're talking about, right? So, we've published research showing that, for example, in the hippocampus, CBD can block a lot of the negative impacts of THC, but that's just looking locally in the hippocampus. But if you look at it from a systemic point of view, if you're giving it like systemically through systemic injections or through edibles, where it's impacting the whole body, then the picture gets way more complicated. And that's something people should keep in mind when sort of reading this literature. You have to consider how localized the effect is. Are you looking at sort of a system-wide effect of these compounds, or are you looking at specific effects in the brain? Because ultimately, how these different circuits interact with each other is what's going to lead to the risk or a reduced risk. And it's hopelessly complex, but ultimately, what we have to do as scientists is sort of adopt a reductionist approach and sort of dissect one circuit at a time. But I would say that, our research has been moving more towards sort of the systemic global approach. So we're doing a lot of edibles. We're doing a lot of, we ultimately want to start moving into vaporized exposure as well, because most people, of course, still inhale cannabis. Edibles are becoming much more popular, but I think, vaping and traditional.
Still the lion's share.
It's still the lion's share for sure. Yeah.
Interesting. So, what are one or two areas that you're actively working on right now that you're particularly excited about? What are some of the questions you're asking?
Yeah, well, I mean, we're continuing with the FABP5 story. So as I said, there's a lot of unanswered questions about that. We want to continue our work into the chronic stress effects and see how the FABP5 FABP5 inhibitors may modulate some of these chronic stress effects. We're doing some PTSD type of studies as well to look at its potential therapeutic impacts for PTSD type disorders. We're continuing our intervention studies, so dietary intervention studies. We're looking now at trying to figure out, and I alluded to this before, how males and females may differ in their sensitivity to these dietary interventions.
Right.
And when is the best time to do the intervention? Obviously, it doesn't, our initial study looked at, and the one that's coming out in a couple of weeks in Molecular Psychiatry, looked at exposure to this dietary intervention at the same time as they were getting the actual THC. But, obviously, given misperceptions around the safety of THC, that's probably not a clinically translatable thing. So most symptoms of serious psychiatric disorders start happening around late adolescence to early adulthood. So what we want to do next is try these dietary interventions during, late adolescence, early adulthood to see if we can prevent the emergence of these symptoms. Sort of after the damage has already been done. Right. So can we somehow mitigate the emergence of those symptoms, similar to that study I was talking about before, the omega-3 study, the Vienna study. And not only are we looking at the omega-3 dietary interventions, but also, the antioxidants as well. So The ones we've looked at so far are L-theanine and acetylcysteine, but there's a host of other ones we can look at as well to see what's most effective.
I mean, I love the idea of exploring whether something like dietary interventions given at a stage of development like late adolescence could work, because, you know, in the real world, for better or for worse, you know, we can't go back in time and erase what happened prenatally or perinatally. But if simple interventions can work before some of these sensitive periods of development have closed and before a lot of these things emerge, like, you know, different forms of psychosis and mood disorders, you know, to the extent that that can be done, that would be a very, very convenient and effective way to, you know, simply through simple means, potentially dietary means, you know, help people.
Exactly. Yeah. And, you know, and that goes on the context of, you know, I mean, the North American diets that we have are horrible, right? So, I mean, there's already existing problems with the type, you know, we tend to have a high omega-6 diet in North America, especially, which I think is leading to a lot of, you know, this disbalance between omega-3s and omega-6s and neuroinflammation. And, you know, there's emerging evidence for that as we move forward. But I think we certainly need to have a serious rethink about the dietary culture that we've sort of created, especially in North America, and how that might be contributing to this mental health epidemic that we have.
No, I mean, I love what you said earlier about, you know, nutritional neuroscience being, you know, a new field. Obviously, nutritional psychiatry is having a bit of a heyday, and you know, a lot of people out there, you know, rightfully so, I think, are focusing on the role of metabolism in psychiatry and mental illness. But one of the things that I think is very destructive and irritating about the way that people tend to ordinarily think about diet and development and health is, I don't know, my whole life growing up, people tended to have the attitude of, oh, when you're young, you can kind of eat anything and your metabolism works. And then it just sort of magically slows down or breaks later. And it's like, no. You are in the process of setting yourself up for it to be broken later by not feeding it properly early on.
Yeah. Oh, absolutely. That's a great point. I mean, look at the junk that so many kids eat. Look, you know, read the ingredients in a box of cereal. I mean, it's just mostly sugar, and it's just it's quite appalling, really. I mean, and part of the problem is that it's hard enough to get kids to eat anything as opposed to nutritional stuff. I struggle with that. I've got a 7 and an 8-year-old, so I appreciate the challenges of getting your kids to eat a healthy diet. But I think that's an excellent point. I think that the problems are probably starting very early in childhood. We're not sort of getting that necessary balance, especially between these fatty acid pathways, particularly omega-6 and omega-3. And that's something that we really need to pay more attention to.
Yeah. And I'm definitely someone who's been harping on the omega-6, the skewed omega-6 composition of diet for a while. Everywhere I look in the literature, whether I'm talking to the cancer people or the brain people like you, when that ratio is skewed, no good seems to come from it. I can't find a clear example where having, 20 to 1 omega-6 to 3 ratio leads to a good outcome.
No, absolutely not. And that's consistent across the, the animal world. I think it's not even just humans, but
Yeah. Is there anything you want to reiterate or anything else you want to mention to people about the general subjects we touched on today before we sign off?
No, I mean, I think I would just reiterate that I think that there's a lot of potential for modulating the FABP system. I think it's we're sort of on the ground floor right now, but as we move forward with different studies, I think obviously I'm biased towards mental health and psychiatric models, but as you mentioned before, I think there's going to be a wide range of applications for modulating the fatty acid binding protein system for cancer, for neurodegenerative disorders, basically anything that might involve some sort of a neuroinflammatory pathology associated with it. So I think it's an exciting time to sort of be looking at these pathways.
All right. Well, Professor Stephen Laviolette, thank you again for joining me again. People who are interested in this stuff, we talked about related stuff, but different stuff in our first episode, and that was definitely one of my favorite ones from that season. So thank you very much.
Thanks for having me. I appreciate it.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.